Good evening, and welcome to Dreamy Academy. Tonight we drift far back through time, not to the birth of stars or drift of continents, but to a universe hidden within us, a realm of profound, intricate beauty, alive with ceaseless activity. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Tonight, we turn our gaze inward to the most intimate universal truth, the cell. It might seem a humble subject, the very foundation of biology, yet within its microscopic boundaries lies a complexity that rivals galaxies, a history that spans billions of years, and a beauty that rewards the most patient contemplation. Right now, as you listen, forces older than mountains are shaping the atoms in your bones, and within every part of your being, tiny, self-contained worlds are performing miracles of chemistry, communication, and self-organization. This isn't just a scientific lecture. It's an invitation to perceive the intricate dance of life at its most fundamental level, to find wonder in the very fabric of your existence. We will unfold understanding like origami in reverse, revealing the elegant truths hidden in plain sight, or rather in plain being. Consider for a moment the ordinary miracle of your own hand, resting quietly as you listen. It seems a solid, continuous thing, a singular entity. Yet, beneath the surface of this apparent stillness, something astonishing is happening. Your hand, like every part of you, is an intricate tapestry woven from countless individual units, each a bustling, self-sufficient world. These are your cells, the fundamental architects of life, the hidden mechanisms that allow you to breathe, to think, to dream. To truly understand ourselves, to grasp the profound interconnectedness of all living things, we must first step into this microscopic realm, a silent, ceaseless ballet of molecules and energy. To begin our journey, let us understand what a cell truly is. Imagine, if you will, not a simple brick in a wall, but a miniature city, complete with its own power plants, transportation networks, communication systems, and even its own recycling centers. Each cell is a distinct, bounded entity, separated from its surroundings by a delicate yet formidable membrane. This membrane is not merely a passive container. It is a dynamic, intelligent frontier, constantly monitoring, communicating, and regulating the flow of life. It is the very first distinction between self and not self, the primordial boundary that made life possible. The concept of the cell itself, so fundamental to our understanding today, was once a profound revelation. It began with the patient gaze of early naturalists, peering through crude lenses. In the 17th century, Robert Hooke, observing a thin slice of corks or tiny box-like compartments, and thinking of a monastery's small rooms, named them. Quote 3. A little later, Antony van Leeuwenhoek, with his superior handcrafted microscopes, turned his gaze to pond water, to his own saliva, and saw, quote, 4 tiny swimming creatures, a vibrant, unseen world teeming with life. These were the first glimpses into the cellular universe, a realization that all living things are composed of these intricate microscopic units. It was a paradigm shift, a quiet opening of a door to an entirely new dimension of existence. At the heart of this cellular existence, this definition of individuality, lies the cell membrane. Picture it not as a rigid wall, but as a shimmering fluid boundary, a delicate film just a few nanometers thick, yet possessing an astonishing complexity. It is primarily composed of a double layer of lipid molecules, tiny fat-like structures, each with a quote 5 that loves water and a quote 6 that shies away from it. These lipids spontaneously arrange themselves into a bilayer, like two sheets of paper pressed together, with their water-loving heads facing outwards, towards the watery environments both inside and outside the cell, and their water-fearing tails tucked safely in the middle. This elegant, self-assembling structure is known as the quote 7, a name that perfectly captures its nature. Fluid, because the lipid molecules can drift and move within the layer, and mosaic, because embedded within this shimmering sea of lipids are various proteins like intricate jewels, each with specific roles. These embedded proteins are the cell's gatekeepers, its sensors, its communicators. Some form channels, allowing specific molecules to pass in and out. Others act as receptors, binding to chemical signals from the outside world, telling the cell what to do, how to react. Still others are enzymes, catalyzing crucial reactions right at the membrane's edge. This membrane, therefore, is not just a barrier. It is a living, breathing interface, a constant conversation between the cell and its environment. It maintains the cell's internal stability, allowing it to create a distinct inner world, a precise chemical environment necessary for life, separate from the often chaotic external conditions. Without this selective permeability, this careful control over what enters and exits, the cell would simply dissolve, its intricate machinery unable to function. There's a particular comfort in understanding that this delicate fluid boundary is the very foundation of all life. From the simplest bacterium to the most complex human being, it's the initial act of creation, the drawing of a line in the primordial soup that allowed for the emergence of organized chemistry, of self-sustaining systems. The cell membrane is the quiet declaration of existence, the first whisper of identity, enabling the internal conditions necessary for the exquisite dance of life to unfold. It is the most fundamental enclosure, allowing for the concentration of molecules, the creation of gradients, and the harnessing of energy, all essential steps towards the vibrant complexity we witness in every living thing. So we've seen how a cell defines its very being by drawing this elegant boundary, a shimmering, intelligent membrane that separates its inner world from the vastness outside. But what truly happens within this enclosed space? What are the intricate components and bustling activities that give this tiny city its life, its purpose, its astonishing capacity for growth, repair, and reproduction? How does this fundamental boundary protect and sustain the molecular marvels it contains? That question leads us deeper into the rich cytoplasm and the specialized organs within, the true heart of cellular life. We left our contemplation at the threshold, didn't we? At that shimmering, intelligent membrane, a boundary both definitive and porous, defining quote 8 and orchestrating the dance of exchange. Now, with a quiet breath, we prepare to step beyond that threshold, to immerse ourselves in the vast, vibrant interior it so carefully protects. Imagine, if you will, gently passing through that lipid bilayer, not dissolving, but simply observing, as if an invisible explorer traversing a newly discovered world. The first sensation, as we drift inward, is one of aqueous expanse. This is the cytoplasm, a magnificent, bustling sea that fills the cell's every available space. It is not merely empty fluid, but a complex, gel-like matrix, teeming with dissolved salts, nutrients, proteins, and the very building blocks of life itself. Think of it as the ocean of this miniature world, a medium through which all the cell's internal components interact, where countless biochemical reactions unfold in a harmonious, ceaseless ballet. It is the stage upon which the grand drama of cellular existence is performed, a dynamic solvent that allows for the intricate choreography of molecules. Here, within this living jelly, the very essence of life's chemistry finds its perfect environment, a delicate balance of viscosity and fluidity, ensuring that molecules can both move freely and yet remain sufficiently organized. And within this vast cytoplasmic ocean, like islands in a vibrant archipelago, reside the organelles, specialized structures, each performing a vital function, each a testament to the cell's profound organizational genius. They are the intricate machinery, the bustling departments of our miniature city, working in concert to sustain life, to respond to stimuli, to grow, and to reproduce. To truly appreciate the cell, we must venture into each of these remarkable entities, understanding their form and their purpose, for together they weave the tapestry of cellular vitality. Our gaze is first drawn, inevitably, to the most prominent of these structures, often positioned at the very heart of the cell, like a serene, spherical command center. This is the nucleus ganda, the cell's vast library and its central processing unit, the very vault of genetic heritage. Encased within its own double membrane, the nuclear envelope, a sophisticated barrier punctuated by pores that carefully regulate traffic, lies the cell's most precious treasure, the DNA. Here, tightly coiled and meticulously organized into chromosomes, resists the complete blueprint for every protein the cell will ever need to construct, every function it will ever perform, every characteristic that defines it. Imagine the nucleus as the ancient sacred archive of a civilization, holding within its walls the complete history, the laws, the engineering schematics, and the artistic inspirations for everything that city is and ever will be. The DNA, those elegant double helices, are the scrolls and codices transcribed and translated with breathtaking precision. From within this hallowed space, instructions are issued, messages dispatched, guiding the cell's growth, metabolism, and reproduction. It is a place of profound quietude, yet immense power, where the very essence of heredity is protected, copied, and expressed. The nucleus is not merely a passive repository. It is an active, intelligent overseer, constantly monitoring the cell's needs, ensuring that the right genes are activated at the right time, a symphony conductor orchestrating the genetic orchestra of life. Its existence underscores a fundamental truth. Life is information, exquisitely stored and meticulously managed. From the nucleus, our attention might drift to the myriad of other structures suspended in the cytoplasm, each a marvel in its own right. Let us turn our focus now to the cell's tireless energy providers, the shimmering bean-shaped powerhouses that fuel every activity, every thought, every beat of your heart. These are the mitochondria. Observe their elegant elongated forms, often in constant motion, dividing and fusing, adapting their shape and number to the cell's energetic demands. Each mitochondrion is a tiny furnace, a miniature power plant with its own distinct inner and outer membranes. The inner membrane, intricately folded into cristae, creates an enormous surface area, a vast landscape where the magic of cellular respiration unfolds. Here, within these folds, glucose and other organic molecules are systematically broken down, not in a sudden burst of combustion, but through a series of controlled, elegant reactions that harness energy to produce adenosine triphosphate, or ATP. Think of ATP as the universal currency of cellular energy. It is the immediate usable form of power that drives muscle contraction, nerve impulses, protein synthesis, and every active transport process. Without the mitochondria, the cell would simply cease to function, a city plunged into darkness, its machinery grinding to a halt. They are the indefatigable workers, tirelessly converting raw materials into the vital energy that sustains every living moment. And what a profound thought that is. These ancient organelles with their own tiny circular DNA, a whisper of their bacterial ancestors, are the very engines that allow us to think, to feel, to dream, to simply be. They connect us through an unbroken chain of energy transformation to the very origins of life on Earth. Moving deeper into the cell's industrial complex, we encounter a vast interconnected network of membranes that permeates much of the cytoplasm. This is the endoplasmic reticulum, ER, a labyrinthine system of flattened sacs called cisternae and branching tubules. It is, in essence, the cell's vast manufacturing and transport facility, a sophisticated internal highway system dedicated to synthesis and movement. The ER comes in two distinct yet interconnected forms. First, there is the rough endoplasmic reticulum, RER, so named because its outer surface is studded with tiny granular structures, the ribosomes. These ribosomes, themselves marvels of molecular engineering, are the cell's protein builders. They read the genetic messages dispatched from the nucleus, messenger RNA, and translate them into long chains of amino acids, the nascent proteins. As these proteins are synthesized on the RER, they are often threaded directly into its lumen, this space within the ER membranes, where they begin their journey of folding into their precise three-dimensional shapes. The RER is particularly abundant in cells that specialize in secreting proteins, such as antibody-producing white blood cells or pancreatic cells that secrete digestive enzymes. It is a protein factory of immense precision and output, where molecular origami is performed with unwavering accuracy. Adjacent to the RER, and often continuous with it, is the smooth endoplasmic reticulum SER. Lacking ribosomes, its surface appears smooth, but its functions are no less vital. The SER is a versatile metabolic workshop. Here, lipids are synthesized, including phospholipids for new membranes and steroids, like the hormones that regulate so many of our bodily functions. It also plays a crucial role in detoxification, metabolizing drugs and harmful substances, particularly abundant in liver cells. Furthermore, the SER acts as a storage site for calcium ions, which are critical for various cellular processes, including muscle contraction. Imagine the SER as the cell's dedicated chemical plant and purification system, constantly working to maintain the cell's internal environment and build essential components. As proteins and lipids emerge from the ER, they are not yet ready for their final destinations. They require further processing, sorting, and packaging, a task expertly handled by the Golgi apparatus, also known as the Golgi complex or Golgi body. This remarkable organelle consists of a stack of flattened membrane-bound sacs, or cisternae, resembling a stack of slightly curved pancakes. It has a distinct polarity, a cis face, which receives vesicles budding off from the ER, and a trans face, from which processed materials exit. Envision the Golgi as the cell's sophisticated postal service and customization shop. As proteins and lipids travel through the Golgi's layers, they undergo modifications. Sugars might be added or removed, specific chemical tags attached, processes that act like molecular zip codes or destination labels. These modifications determine a molecule's final destinations, whether they will be secreted outside the cell, integrated into the plasma membrane, or delivered to other organelles like lysosomes. Finally, at the trans face, these perfectly modified and sorted molecules are packaged into new vesicles, which then bud off and travel to their designated locations. The Golgi apparatus represents an astonishing level of organization, ensuring that every molecular package arrives at its correct address, a testament to the cell's unwavering commitment to order and efficiency. And what of those tiny ubiquitous ribosomes we glimpsed on the RER? While many are attached to the ER, many more float freely in the cytoplasm. These are the fundamental workhorses of protein synthesis, the molecular machines that read the mRNA instructions and assemble amino acids into proteins. They are found in every living cell, from the simplest bacteria to the most complex human cells, a universal testament to the centrality of proteins in all life. Imagine them as countless diligent construction workers, each focused on building a specific component according to the master plan, tirelessly contributing to the cell's structure and function. Now, every city needs a waste management and recycling system, and our cellular metropolis is no exception. This critical function is performed by the lysosomes and peroxisomes. Lysosomes, often described as the cell's recycling centers or digestive organs, are membrane-bound sacs containing powerful hydrolytic enzymes. These enzymes are capable of breaking down worn-out organelles, cellular debris, and foreign particles ingested by the cell. They are the diligent custodians ensuring that cellular waste is efficiently broken down and its valuable components recycled, preventing harmful accumulation. Without lysosomes, cells would become clogged with debris, unable to function. Peroxisomes, smaller and structurally simpler than lysosomes, are equally vital. They specialize in detoxification, containing enzymes that neutralize harmful byproducts of metabolism, such as hydrogen peroxide. They are particularly active in liver and kidney cells, where they help to detoxify alcohol and other harmful compounds. Think of peroxisomes as the cell's specialized hazardous waste disposal units, protecting the cell from internal pollutants and maintaining a pristine internal environment. Finally, let us consider the cell's internal scaffolding, its dynamic architecture, and its intricate transportation network, the cytoskeleton. This is not a static framework, but a constantly remodeling lattice of protein filaments that extends throughout the cytoplasm. It is composed of three main types of fibers: microtubules, intermediate filaments, and microfilaments. The cytoskeleton provides structural support, maintaining the cell's shape and resisting mechanical stress, much like the steel girders of a skyscraper. But its role extends far beyond mere scaffolding. It is also responsible for cell movement, enabling cells to crawl, change shape, and divide. It provides the tracks along which organelles and vesicles are transported, a bustling internal railway system where molecular motors carry their cargo to precise destinations. Imagine the cytoskeleton as the cell's dynamic architectural firm, constantly redesigning, building, and moving, ensuring both stability and adaptability. It is a silent, ceaseless ballet of assembly and disassembly, reflecting the cell's incredible capacity for internal organization and responsiveness. As we gently pull back from this micro-exploration, allowing the full panorama of the cell's interior to re-emerge, we are left with a profound sense of awe. We have journeyed through the cytoplasm, the bustling aqueous medium, and explored some of its most remarkable inhabitants. The nucleus, the silent archive of life, the mitochondria, the tireless generators of energy, the endoplasmic reticulum, the vast factory and transport hub, the Golgi apparatus, the meticulous sorter and packager, the ribosomes, the universal builders, the lysosomes and peroxisomes, the diligent custodians, and the cytoskeleton, the dynamic architect and mover. Each organelle, with its specialized structure and function, is a masterpiece of biological engineering. Yet, it is their collective, integrated action, their seamless communication and cooperation, that transforms a mere collection of molecules into a living, breathing, self-sustaining entity. The cell is indeed a miniature city, but one far more complex, far more efficient, and infinitely more awe-inspiring than any human construct. It is a universe of intricate beauty, operating with a precision that rivals the most sophisticated clockwork, yet imbued with a flexibility that allows it to adapt and thrive. Our journey continues, for even this glimpse only scratches the surface of the cell's profound mysteries. We have seen the major players, but the interactions, the regulatory networks, the signaling pathways that connect them all, these are the next layers of wonder we shall gently unfold. For now, let us simply hold this image of the bustling, brilliant interior, a testament to life's fundamental elegance and power. Having journeyed through the intricate architecture of the cell, exploring its shimmering outer boundary and the bustling metropolis of its internal organelles, we now arrive at a profound realization. A cell is not merely a collection of specialized compartments. No, it is far more. It is a living, breathing symphony, a highly coordinated entity where every component plays a vital role in a ceaseless dance of interaction and communication. Imagine, if you will, the most sophisticated city you can conceive, a place where every citizen, every building, every system is not only perfectly engineered but also intrinsically linked, constantly exchanging information, adapting and responding to the world around it. This is the cell. To truly appreciate the breathtaking complexity and elegance of this miniature universe, we must now delve into the very language it speaks, the intricate pathways of cellular communication. How do these myriad organelles, these countless proteins and molecules, know what to do, when to act, and how to coordinate their efforts? How does a cell perceive its environment, sense danger, or receive instructions from its neighbours? This is where the magic of signalling pathways unfolds, revealing a system of astonishing precision and adaptability. Consider for a moment the vast, silent spaces between cells in your own body. They are anything but silent. They are teeming with molecular whispers, with chemical signals travelling through the extracellular fluid, carrying messages of growth, repair, defense, and specialization. A cell, you see, is never truly isolated. It is a social being, constantly listening, interpreting, and responding to the chorus of its surroundings. At the heart of this communication lies a fundamental principle: reception, transduction, and response. It begins with a signal, often a specific molecule known as a ligand. Think of a ligand as a key, crafted with exquisite specificity to fit a particular lock. This lock is a receptor, a protein molecule typically embedded within the cell membrane or residing within the cytoplasm. When the ligand binds to its complementary receptor, it triggers a conformational change, a subtle shift in the receptor's shape, much like the turning of a key in a lock. This initial binding event is the moment of reception. This change in the receptor's shape isn't just a static event. It initiates a cascade of molecular interactions within the cell. This is the process of transduction, where the external signal is converted into an internal cellular response. Imagine a domino effect, but one of immense sophistication and branching pathways. One molecule activates another, which activates another, often amplifying the signal along the way. These internal messengers, sometimes called second messengers, like cyclic AMP or calcium ions, spread the message rapidly throughout the cell, reaching various target proteins. This intricate relay system ensures that a single external signal can provoke a complex and multifaceted internal reaction. Finally, this internal message culminates in a response. This response can take countless forms. Perhaps the cell begins to synthesize new proteins, or it changes its metabolic activity, or it alters its shape, or it prepares to divide. It might even trigger a programmed self-destruction if it's a damaged or unwanted cell, a process known as apoptosis. The cellular response is always precisely tailored to the original signal, a testament to the cell's inherent intelligence and its ability to maintain delicate internal balance. This language of ligands and receptors, of cascades and responses, is not just about individual cells reacting to their immediate environment. It orchestrates the grander tapestry of multicellular life. Paracrine signaling, for instance, involves local communication, where cells secrete substances that act on nearby cells. Think of a neighborhood chat. Endocrine signaling, on the other hand, is like a global broadcast, where hormones are released into the bloodstream to travel to distant target cells throughout the body. And autocrine signaling is a cell talking to itself, secreting signals that bind to its own receptors, often used in development or in fine-tuning responses. Even direct contact between cells, through specialized junctions, allows for the intimate sharing of information. As we ponder this elaborate dance of molecular communication, we cannot help but feel a profound sense of awe. This is the very essence of life's responsiveness, the dynamic interplay that allows an organism to grow, heal, adapt, and survive. It is a universal language, spoken by every living cell, from the simplest bacterium to the most complex neuron in your brain. And what is one of the most fundamental responses a cell can enact? The perpetuation of its own existence, the creation of new life. This brings us to another magnificent choreography within the cellular realm, the cell cycle and cellular division. Just as a great story demands to be told again and again, ensuring its legacy, so too does a cell embark on a meticulously orchestrated journey to replicate itself, passing on its precious genetic blueprint to a new generation. The cell cycle is a continuous, tightly regulated sequence of events that encompasses growth, DNA replication, and cell division. It is a testament to life's relentless drive for continuity. We can broadly divide this cycle into two main phases: interphase and the M phase, mitotic phase. Interphase is often mistakenly thought of as a resting phase, but nothing could be further from the truth. It is a period of intense activity, growth, and preparation, where the cell meticulously duplicates its contents, ensuring that each daughter cell receives a complete set. Interphase itself is subdivided into three distinct stages. First, we have G1 phase, gap 1. This is a period of significant cellular growth and normal metabolic activity. The cell is busy, synthesizing proteins, expanding its cytoplasm, and generally getting ready for the monumental task ahead. It's a time for assessment, for checking if conditions are favorable for division. Next comes the pivotal S phase, synthesis phase. This is where the cell's most precious cargo, its DNA, is faithfully replicated. Imagine, if you will, these cells' entire library of genetic instructions being meticulously copied, word for word, chapter for chapter, ensuring that not a single piece of information is lost or corrupted. This process is astonishingly accurate, a testament to the sophisticated molecular machinery that unwinds the double helix, synthesizes new complementary strands, and then rewinds them. By the end of S phase, each chromosome consists of two identical sister chromatids, joined at a central point called the centromere. The cell has effectively doubled its genetic material. Following this crucial duplication, we enter the G2 phase, gap 2. Here, the cell continues to grow, synthesizes proteins and organelles necessary for division, and meticulously checks for any errors in the duplicated DNA. It's a final preparation stage, ensuring that everything is in perfect order before the grand performance of division begins. Once interphase is complete, the cell is ready for the dramatic climax, the SAPUM phase, which includes mitosis, nuclear division, and cytokinesis, cytoplasmic division. Mitosis itself is a continuous process, but for ease of understanding, we describe it in 4 distinct subphases, each a marvel of cellular choreography. In prophase, the long, diffuse strands of chromatin in the nucleus condense and become visible as distinct chromosomes. The nuclear envelope begins to break down, and the mitotic spindle, a framework of microtubules, starts to form from these entrosomes, which have moved to opposite poles of the cell. As we move into metaphase, the chromosomes, now fully condensed and distinct, align themselves precisely along the cell's equatorial plate, an imaginary line equidistant from the 2 spindle poles. Each sister chromatid is attached to spindle fibers from opposite poles, poised for separation. This alignment is critical, ensuring that each new cell receives an identical set of chromosomes. Then, with breathtaking precision, comes anaphase. The sister chromatids suddenly separate, pulled apart by the shortening spindle fibers towards opposite poles of the cell. Each chromatid is now considered a full-fledged chromosome. This is a moment of profound significance, the physical division of the genetic material. Finally, in telophase, the separated chromosomes arrive at the poles and begin to decondense. New nuclear envelopes form around the 2 sets of chromosomes, and the mitotic spindle disassembles. The cell now contains 2 distinct nuclei, each with a complete and identical set of genetic information. Concurrent with or immediately following mitosis is cytokinesis, the division of the cytoplasm. In animal cells, a contractile ring of actin and myosin filaments forms just inside the cell membrane along the metaphase plate, pinching the cell into two separate daughter cells. Each new cell receives a full complement of cytoplasm, organelles, and, crucially, an identical set of chromosomes. This entire process from G1 through cytokinesis is meticulously regulated by a series of internal ABERC checkpoints. These checkpoints act as surveillance mechanisms, ensuring that each step is completed accurately and that the cell is ready to proceed to the next phase. If errors are detected, this cell cycle can be paused, allowing for repair, or the cell can be directed towards apoptosis, preventing the propagation of damaged or potentially harmful cells. This intricate control system is vital, for uncontrolled cell division is, at its heart, what defines cancer. The beauty of the cell cycle lies not only in its precision but in its profound implications. It is the mechanism by which life renews itself, by which organisms grow, by which tissues are repaired, and by which the genetic legacy is passed from one generation of cells to the next. Every cell in your body, with very few exceptions, has arisen from a long, unbroken chain of cell division stretching back billions of years. It is the very engine of continuity. And what happens after division, when a new cell is born? Does it simply continue to divide indefinitely? Sometimes a yes. But often, especially in complex multicellular organisms like ourselves, these new cells embark on a journey of specialization. This phenomenon is known as cellular differentiation. From a single fertilized egg, a process of repeated division and differentiation gives rise to the hundreds of distinct cell types that constitute a human body. Nerve cells, muscle cells, skin cells, blood cells, each with a unique structure and function, yet all containing the same genetic blueprint. It is a testament to the cell's ability to interpret its genetic instructions in a context-dependent manner, activating some genes and silencing others to adopt a specific identity and role within the grand organismal design. Imagine our miniature city, not just replicating its general form, but creating specialized districts with unique architectures and purposes, all from the same initial blueprint. Ultimately, all these processes, communication, division, differentiation, contribute to a cell's overarching goal, the maintenance of homeostasis. Homeostasis is the dynamic equilibrium, the constant effort to maintain a stable internal environment despite fluctuations in the external world. It is the cell's unwavering commitment to balance, to optimal function. Through its intricate signaling networks, its controlled growth and division, and its specialized functions, the cell continuously monitors and adjusts, a self-regulating entity striving for perfect harmony. As we conclude this chapter, reflect on the extraordinary intelligence embedded within every cell. It communicates with exquisite precision, replicates its entire being with astounding fidelity, and specializes to contribute to a larger whole, all while tirelessly maintaining its internal balance. The cell is not a passive bag of chemicals. It is an active, intelligent, and profoundly beautiful manifestation of life itself. And in understanding its fundamentals, we begin to truly grasp the wonder of our own existence. We stand at the threshold of ever deeper insights, ready to explore the even more intricate regulatory networks and energetic exchanges that define this marvelous frontier. Having journeyed through the cell's dynamic boundaries, its bustling internal landscape, and the intricate symphony of its communication and replication, we now turn our gaze to the very heartbeat of its existence, the ceaseless flow of energy that powers every whisper, every movement, every thought within its microscopic walls. We spoke of mitochondria as the cell's powerhouses, the generators of ATP, the universal energy currency. But how precisely is this currency minted? How does the cell orchestrate such a magnificent, continuous energetic exchange, sustaining its myriad functions with breathtaking efficiency? Imagine, if you will, the cell not just as a miniature city, but as a living, breathing economy, constantly balancing its budget of resources and energy. This is the realm of cellular metabolism, a vast, interconnected network of biochemical reactions that build up anabolism and break down catabolism molecules to sustain life. It is an intricate dance of atoms and electrons, a testament to nature's profound ingenuity. Let us begin our exploration with the grand pathways of energy generation, the catabolic processes that dismantle fuel molecules to release their stored potential. The primary fuel for many cells is glucose, a simple sugar molecule, a concentrated packet of solar energy, originally captured by plants through photosynthesis and now ready to be unleashed. Our journey into energy extraction commences in the cytoplasm, the very heart of the cell's aqueous environment, with a process known as glycolysis. The name itself, sugar splitting, tells a precise story. Here, in a series of 10 enzymatic steps, a single molecule of glucose, a 6-carbon ring, is meticulously cleaved and rearranged. It's a process that doesn't require oxygen, making it an ancient and universal pathway, a fundamental energy-generating mechanism conserved across nearly all life forms. As glucose breaks down, it yields 2 molecules of pyruvate, a 3-carbon compound. But more crucially, in this initial burst of activity, a small, yet significant amount of ATP is directly produced, akin to finding a few coins in your pocket. Furthermore, electrons are harvested and loaded onto an electron carrier molecule called NADH, like small energy batteries being charged, ready for a much larger payoff later. From this cytoplasmic prelude, the pyruvate molecules, now holding much of the glucose's remaining energy, embark on a deeper journey. For cells operating under aerobic conditions, that is, in the presence of life-giving oxygen, pyruvate migrates into the inner sanctum of the mitochondria, those oval-shaped powerhouses we first encountered. Here, within the mitochondrial matrix, a new chapter of energy extraction unfolds. Each pyruvate molecule undergoes a transformation, losing a carbon atom as carbon dioxide, a metabolic sigh of the cell, and becoming an acetyl group. This acetyl group then attaches to a coenzyme, forming acetyl-CoA, the entry point into the next major stage, the Bas-Krebs cycle, also known as the citric acid cycle. The Krebs cycle is a marvel of cyclical biochemistry, a metabolic carousel that spins twice for each original glucose molecule, once for each acetyl-CoA. Imagine a meticulously choreographed molecular ballet, where the acetyl group enters a 4-carbon compound forming citrate, then undergoes a series of transformations, releasing more carbon dioxide and, most importantly, generating a substantial number of additional electron carriers, NADH and FADH2. A small amount of ATP is also directly produced here, But the true wealth of the Krebs cycle lies in these charged electron carriers, now brimming with high-energy electrons poised to unleash their power. It is a process of elegant, systematic dismantling, extracting every last bit of usable energy from the original glucose molecule, preparing it for the grand finale. This finale, the crescendo of cellular respiration, is known as oxidative phosphorylation, and it occurs along the intricate folds of the inner mitochondrial membrane. This is where the vast majority of the cell's ATP is generated, a process so fundamental and so beautiful that it often evokes a sense of profound wonder. Here, the NADH and FADH2, laden with their high-energy electrons, arrive like vital messengers. They deliver their electrons to a series of protein complexes embedded within the membrane, collectively known as the electron transport chain. Envision this chain as a molecular cascade, a series of stepping stones down which electrons fall, releasing energy at each step, much like water tumbling down a waterfall. This released energy is not immediately converted into ATP. Instead, it is masterfully harnessed to pump protons, hydrogen ions, from the mitochondrial matrix into the intermembrane space, creating a steep electrochemical gradient. This gradient is a form of stored potential energy, akin to water held behind a dam. The protons, driven by this gradient, then rush back across the membrane, but not just anywhere. They flow through a remarkable molecular rotary motor called ATP synthase. ATP synthase is an enzyme of extraordinary design, a true molecular marvel. As protons stream through it, they cause a part of the enzyme to spin, much like a turbine. This mechanical rotation drives the synthesis of ATP from ADP and inorganic phosphate. It is here, through this elegant mechanism of chemiosmosis, that the cell produces a torrent of ATP, literally powering life itself. And what happens to the electrons after their journey down the transport chain? They are finally accepted by oxygen, which combines with protons to form water, a simple yet utterly essential byproduct of this entire process. Without oxygen, this final step cannot occur. The electron transport chain grinds to a halt, and the vast majority of ATP production ceases, underscoring the vital role of the air we breathe. This catabolic journey, from glucose to carbon dioxide and water, culminates in the magnificent production of ATP, the energetic currency that fuels every cellular endeavor we have discussed. From membrane transport and protein synthesis to cellular division and communication. But metabolism is not solely about breaking down. It is also about building up. Anabolism. The constructive phase of metabolism utilizes this hard-earned ATP to synthesize the complex molecules necessary for cell structure, function, and growth. The very intermediates generated during glycolysis and the Krebs cycle are not merely waste products. They are often diverted and repurposed as building blocks. Imagine pyruvate being converted into amino acids, or acetyl-CoA serving as a precursor for fatty acids and lipids. The metabolic pathways are not isolated linear roads, but an intricate interwoven web, a sophisticated network where molecules can be channeled in multiple directions depending on the cell's immediate needs. This metabolic flexibility allows the cell to adapt, to grow, to repair itself, and to respond to an ever-changing environment, whether it's a feast of nutrients or a period of scarcity. Of course, such a complex and vital system cannot operate unchecked. The cell maintains an exquisite level of regulation and control over its metabolic pathways, This is where the intricate regulatory networks hinted at in our previous discussions come into full view. Enzymes, those molecular catalysts that accelerate biochemical reactions, are key players in this orchestration. Their activity can be finely tuned. They can be activated or inhibited, often by the very products of the pathways they govern, a principle known as feedback inhibition or activation. If a cell has enough ATP, for instance, the enzymes involved in ATP synthesis might be temporarily slowed down. Conversely, if energy levels are low, these pathways are vigorously stimulated. Beyond this internal feedback, external signals, the very same ligands and receptors we explored in cellular communication, play a profound role. Hormones like insulin, signaling an abundance of glucose, trigger cells to increase glucose uptake and storage. Conversely, glucagon signals glucose scarcity, prompting cells to release stored energy. These are not merely isolated signaling events. They are directives that ripple through the metabolic landscape, adjusting the rates of glycolysis, gluconeogenesis, glucose synthesis, and glycogen storage, ensuring that the cell, and indeed the entire organism, maintains a stable and healthy internal environment, that crucial state of homeostasis. To truly appreciate the cell, therefore, is to understand this incessant, elegant flow of energy. It is to marvel at the molecular machinery that transforms the potential energy locked in chemical bonds into the kinetic energy of life. Every beat of your heart, every thought that flickers through your mind, every intricate movement of your hand as you ponder these words is powered by countless trillions of these minute, yet magnificent energetic exchanges occurring within the cells of your body. We have traversed the fundamental landscape of the cell, from its outer membrane to its internal organelles, from its communication networks to its cycles of division, and now to the very engine of its existence, its metabolism. Yet, even as we grasp these foundational truths, the universe hidden within us continues to unfold, revealing even more intricate layers of regulatory networks, the subtle whispers of genetics, and the profound implications for health and disease. This journey of discovery, like the metabolic pathways themselves, is continuous, ever deepening, and perpetually awe-inspiring. We stand at the threshold of further wonders, ready to delve into the very blueprint that guides these intricate processes, the genetic information that defines a cell, and indeed defines life itself. We have journeyed through the dynamic landscape of the cell, observing its intricate boundaries, its bustling internal cities, its relentless pursuit of energy, and its profound capacity for communication and self-renewal. We've seen how every process, from the whisper of a signal across a membrane to the furious churn of metabolic pathways, contributes to the grand symphony of life. Yet beneath all this ceaseless activity, within the very heart of the cellular command center, lies the master plan, the foundational scroll upon which all instructions for existence are etched. This, my friends, is the realm of genetics, the subtle whispers that dictate the very essence of who and what a cell is, and by extension who and what you are. Imagine, if you will, that the cell is not merely a miniature city, but an ancient, sprawling library filled with countless volumes detailing every aspect of its construction, operation, and potential. Within the hushed sanctum of the nucleus, this library's most sacred and vital tome resides, the deoxyribonucleic acid, or DNA. It is not just a molecule, It is the inherited scroll of life, a profound testament to continuity and identity, passed down through generations, carrying the echoes of ancestral beginnings and the blueprint for future possibilities. To truly appreciate this marvel, let us first gaze upon its structure. If we could shrink ourselves down to navigate this molecular architecture, we would find DNA not as a flat blueprint, but as a majestic spiraling staircase, a double helix. Picture two elegant strands, intertwined like a delicate ladder twisted upon itself, ascending into an unseen infinity. Each rung of this ladder is formed by a pair of chemical units called nitrogenous bases, always pairing in a precise, almost poetic dance. Adenine, A, always with thymine, T, and cytosine, C, always with guanine, G. These are the fundamental letters of life's alphabet, arranged in sequences of breathtaking complexity. The sides of this ladder, the sugar-phosphate backbone, provide the sturdy support, a continuous chain that holds the precious sequence of bases in perfect order. This elegant, predictable pairing is not merely a structural detail, it is the very foundation of how genetic information can be faithfully copied and precisely interpreted. Within this colossal molecular library, specific chapters or paragraphs are known as genes. Each gene is a segment of DNA, a particular sequence of these A, T, C, and G letters, which holds the instructions for building a specific product, most often a protein. Think of your own hand, the intricate network of bones, muscles, and nerves that allows you to grasp, to feel, to write. Every single protein required for the formation and function of that hand, from the collagen that gives your skin elasticity to the enzymes that facilitate muscle contraction, has its origin in a specific gene, a particular set of instructions within your DNA. It is a universe of information, compressed into a microscopic scale, yet dictating the macroscopic reality of your being. The profound responsibility of DNA is not just to store this information, but to ensure its perfect transmission. When a cell prepares to divide to create two new daughter cells, each must receive an identical, complete copy of this master blueprint. This astonishing feat is accomplished through a process called DNA replication. Imagine the entire double helix gently unwinding, much like unzipping a zipper, separating the two intertwined strands. Each separated strand then acts as a template, a guide for building a new complementary partner. Following the strict rules of base pairing, A with T, C with G, new nucleotides, the building blocks of DNA, are meticulously added to each template strand. Enzymes, like diligent molecular architects, orchestrate this symphony of synthesis, ensuring that each new strand is a perfect mirror image of its original partner. The result is two identical double helices, each containing one original strand and one newly synthesized strand. It is a breathtaking testament to molecular precision, a process that ensures the continuity of life, from a single cell to the vast biodiversity of our planet, preserving the genetic legacy with astonishing fidelity. Yet DNA, this precious master blueprint, generally remains safely sequestered within the nucleus, protected from the bustling activity of the cytoplasm. How then do its instructions, its genes, translate into the dynamic functional proteins that power the cell? This is where our story introduces another crucial player, ribonucleic acid, or RNA. If DNA is the master library, RNA is the diligent scribe, the messenger, and the versatile artisan, carrying copies of specific instructions out into the cellular factory to oversee their execution. This journey from gene to protein, this fundamental flow of genetic information, is often referred to as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. It is a concept of elegant simplicity, yet it underpins all of life's complexity. The first step in this transformative journey is transcription. Here, a specific segment of DNA, a gene, is temporarily unwound. Instead of creating a new DNA strand, an enzyme called RNA polymerase synthesizes a complementary strand of RNA from one of the DNA templates. This new RNA molecule, often a messenger RNA, mRNA, carries the specific genetic message from the gene. It's like photocopying a single recipe from the master cookbook, a temporary working copy that can be taken to the kitchen. Notably, in RNA, the base thymine, T, is replaced by uracil, U, adding a subtle but distinct difference to its chemical signature. Once transcribed, this RNA molecule, carrying its precious instructions, is free to leave the protective confines of the nucleus and venture into the cytoplasm. There, in the cytoplasm, or sometimes attached to the endoplasmic reticulum, the mRNA meets the cell's protein-building factors, the ribosomes. This is where the second, equally astounding step, translation, unfolds. Imagine the mRNA strand as a long ribbon of code, a sequence of three-letter words. Each of these three-letter words is called a codon, and each codon specifies a particular amino acid, the building blocks of proteins. The ribosome, like a skilled reader, moves along the mRNA, interpreting each codon. As the ribosome reads, specialized RNA molecules called transfer RNA, tRNA, act as molecular adapters. Each tRNA molecule has a specific three-letter anticodon that is complementary to an mRNA codon, and it also carries a specific amino acid. When a tRNA's anticodon matches an mRNA codon on the ribosome, it delivers its amino acid to the growing protein chain. One by one, amino acids are linked together by peptide bonds, forming a nascent protein, folding into its unique three-dimensional shape, ready to perform its specific function. It is a ballet of molecular recognition and precise assembly, orchestrated with breathtaking speed and accuracy, transforming a linear code into a functional, dynamic molecular machine. These proteins, once assembled, are the true workhorses of the cell. They are the enzymes that catalyze every metabolic reaction, the structural components that give cells their shape, the transporters that move molecules across membranes, the receptors that receive signals, and the motors that drive cellular movement. They are the very essence of cellular activity, and their diversity of form and function is astounding, all dictated by the precise sequence encoded in your DNA. But what happens when there's a typo in the master blueprint? These changes in the DNA sequence are known as mutations. Sometimes, a single base pair might be substituted, inserted, or deleted. These alterations can arise spontaneously during DNA replication, or they can be induced by external factors like radiation or certain chemicals. The consequences of a mutation can vary widely. Some are silent, having no effect on the resulting protein. Others might be beneficial, leading to new traits that aid adaptation and drive evolution. Yet many mutations can be detrimental, altering a protein's function, leading to cellular dysfunction and manifesting as various diseases including cancer. The cell has elaborate repair mechanisms to correct these errors, but they are not infallible. The delicate balance between stability and change is a constant theme in the genetic story. And finally, to truly grasp the profound elegance of this system, we must acknowledge that not all genes are on all the time. Imagine our library again. You don't need every book open on every table simultaneously. Different cells, despite containing the identical genetic blueprint, express different sets of genes. A skin cell functions differently from a neuron, not because it has different genes, but because it expresses different genes. This intricate control mechanism, known as gene regulation, determines which genes are transcribed into RNA and translated into protein, and when, where, and to what extent. It involves a sophisticated network of molecular switches, activators, and repressors, ensuring that each cell performs its specialized role within the complex tapestry of a multicellular organism. It is the ultimate orchestration, allowing a single blueprint to give rise to the astounding diversity of cell types and functions that constitute a living being. As we conclude this chapter, reflect on the sheer wonder contained within the double helix. Within your own body, in every one of your trillions of cells, this intricate dance of DNA, RNA, and protein unfolds ceaselessly. It is the very language of life, the code that defines your individuality, the legacy that connects you to all living things, and the foundation upon which all cellular processes, from metabolism to communication, are built. The genetic blueprint is not a static artifact. It is a living, breathing set of instructions, constantly being read, copied, and regulated, shaping the very essence of existence. It is here, in these subtle whispers of genetics, that the universe truly begins to articulate itself within us, in a language of unparalleled beauty and power. In our last shared exploration, we journeyed deep into the very blueprint of life itself, unraveling the elegant double helix of DNA, witnessing the intricate dance of transcription and translation, and marveling at the masterful orchestration of gene regulation. We saw how the selective expression of genes allows a single and primordial cell to diversify, to specialize, to become a neuron, a muscle fiber, a skin cell, each a unique instrument in life's grand orchestra, playing its distinct part. But what happens when these specialized instruments begin to play together? How do these millions, billions, trillions of unique cellular entities coalesce into something far greater than the sum of their individual parts? How do they construct the magnificent self-sustaining marvel that is you? Today, we extend our gaze, moving beyond the singular self-sufficient city of the cell to observe how these intricate units collaborate, forming the breathtaking complexity of a multicellular organism. Imagine, if you will, not just one miniature city, but an entire planet teeming with diverse metropolises, each connected, each contributing to the vitality of the whole. This is the essence of multicellularity, a profound leap in biological organization, where cooperation becomes the bedrock of existence. Our journey begins with the concept of tissues, as individual cells specialize through the exquisite choreography of gene regulation. They don't merely exist in isolation. Instead, cells with similar structures and functions gather, bind together, and work in concert, forming distinct functional groups. Think of them as the foundational building blocks, the primary sections of our biological symphony. We encounter, for instance, epithelial tissue, a shimmering protective layer that covers surfaces, lines cavities, and forms glands. Your skin is a testament to its protective embrace, a living shield against the world. Then there is connective tissue, the versatile scaffolding and binding agent, providing support, connecting disparate parts, and even storing energy. From the fibrous tendons that link muscle to bone, to the adipose tissue that cushions and insulates, connective tissue is the silent architect, holding everything in its proper place. We also discover muscle tissue, the engine of movement, capable of contraction and relaxation, powering every beat of your heart, every breath you take, every step you embark upon. And finally, the electrifying network of nervous tissue, a marvel of rapid communication, weaving thoughts, sensations and commands into the very fabric of your being. It is a biological internet, transmitting signals with astonishing speed and precision. These tissues, each a mosaic of specialized cells, do not operate in isolation. No, they are called upon to combine, to integrate, to form structures of even greater complexity. The organs. An organ is a discrete structure composed of two or more different tissue types, working together to perform a specific vital function. Consider your heart, a tireless pump, a testament to coordinated effort. It comprises muscle tissue for contraction, nervous tissue to regulate its rhythm, connective tissue for structural integrity, and epithelial tissue lining its chambers. Each tissue plays its part, contributing to the heart's singular purpose, to circulate life-sustaining blood throughout your entire being. Or ponder the intricate architecture of your brain, where billions of neurons supported by a vast network of glial cells, a type of connective tissue, weave the tapestry of consciousness, memory and emotion. Each organ is a masterpiece of biological engineering, a testament to the power of integration. And the story continues to unfold, revealing yet another layer of organization. Organs, in turn, do not work in isolation. They form organ systems, groups of organs that cooperate to perform major functions for the body. The circulatory system, with the heart, blood vessels and blood, transports nutrients, oxygen and waste. The respiratory system, with the lungs and airways, facilitates the exchange of gases. The digestive system, a winding pathway from mouth to intestine, breaks down food and absorbs nourishment. The nervous system, a central command center, coordinates all these activities, allowing you to perceive, react, and interact with your world. Each system is a grand alliance, a sophisticated network of specialized organs, all working in a harmonious, ceaseless ballet to sustain life. This journey from cell to tissue to organ system, and finally to the complete organism, reveals a profound truth. Life at every level is about interconnectedness. The specialized functions we observed in individual cells, the energy production of mitochondria, the protein synthesis of ribosomes, the signaling pathways of receptors, now take on a new, grander significance when viewed through the lens of multicellularity. Each cellular activity, each metabolic pathway, each gene expressed or silenced, contributes to the overall well-being of the organism. This brings us back to a concept we touched upon earlier, now amplified to an organismal scale: homeostasis. If within a single cell, homeostasis is the maintenance of a stable internal environment, then within a multicellular organism, it is a magnificent dynamic orchestration, a constant dance of regulation to maintain optimal conditions for all its billions of cells. Your body temperature, blood sugar levels, pH balance, oxygen saturation, These are not static values, but rather exquisitely regulated variables, constantly monitored and adjusted through an intricate web of feedback loops. When you shiver in the cold, your muscle cells generate heat. When your blood sugar rises, pancreatic cells release insulin to prompt other cells to absorb glucose. These are not isolated events. They are coordinated responses involving multiple organ systems, all striving to restore and maintain the delicate equilibrium essential for life. It is a testament to the evolutionary genius woven into our very being, a living, breathing testament to resilience and adaptation. But what happens, dear seeker, when this intricate dance falters? What occurs when the symphony loses its rhythm, when the finely tuned mechanisms of homeostasis begin to fray? Here we enter the realm of disease, a disruption of the harmonious balance that sustains life. Disease, in essence, is a failure of cellular and organismal homeostasis, a breakdown in the communication, regulation, or function of our biological systems. Consider, for instance, the insidious nature of cancer. We spoke of the meticulous checkpoints that govern the cell cycle, ensuring flawless replication and division. Cancer cells are, in many ways, cells that have forgotten their place in the grand design. Through mutations, alterations in the genetic blueprint we explored, these cells lose their ability to respond to regulatory signals. They ignore the commands to stop dividing, to undergo programmed cell death, apoptosis, when damaged, or to remain confined to their designated tissues. They become rogue elements, replicating uncontrollably, consuming resources, and disrupting the function of surrounding healthy cells and tissues. It is a profound failure of cellular self-control and intercellular communication, a stark reminder of the delicate balance required for ordered growth. Then there are infectious diseases, where external invaders, bacteria, viruses, fungi, parasites, breach our protective barriers and hijack our cellular machinery. A virus, for example, is little more than a genetic package, often encased in protein, that invades a host cell, reprogramming it to produce more viral particles, ultimately disrupting the cell's normal functions and often leading to its demise. Our immune system, a complex network of specialized cells we will explore more deeply another time, is our valiant defense, a testament to the body's ability to identify and neutralize threats, but even it can be overwhelmed. We also face genetic disorders. Conditions arising directly from errors in our DNA, the very code of life. A single, misplaced base pair, a missing gene, or an extra chromosome can lead to faulty proteins, dysfunctional enzymes, or impaired regulatory pathways, manifesting as diseases that affect cellular function at its most fundamental level. Cystic fibrosis, for example, results from a mutation in a gene encoding a protein channel crucial for chloride transport, leading to thick, sticky mucus buildup in various organs. These are whispers from the blueprint itself, profound reminders of the precision required for life to flourish. And finally, there is the inexorable march of aging. While not a disease in itself, aging represents a gradual decline in the efficiency of cellular and organismal homeostasis. Cells accumulate damage over time. Their ability to repair themselves diminishes, telomeres shorten, and mitochondria become less efficient. The intricate regulatory networks that once functioned flawlessly begin to falter, making the organism more susceptible to disease and less capable of maintaining its internal equilibrium. It is a slow, graceful descent, a testament to the transient nature of even the most magnificent biological machines. Yet, in understanding these disruptions, we find not despair, but profound hope. For every revelation about the inner workings of the cell, for every layer of complexity we peel back, we gain new insights into preventing, treating, and even curing disease. The knowledge we glean from cell biology is the bedrock of modern medicine. It drives the development of new drugs that target specific cellular pathways, informs the design of gene therapies that correct genetic errors, and fuels the promise of regenerative medicine, where stem cells might one day repair damaged tissues and organs. It is the foundation for personalized medicine, tailoring treatments to the unique cellular landscape of each individual. Our journey through the fundamental principles of cell biology is far from over. Each chapter, each concept, unfurls like a petal on an infinitely complex flower, revealing ever more intricate beauty. We began with the cell's boundary, journeyed through its bustling interior, explored its communication and reproduction, traced its energy flows, and deciphered its genetic code. Now, we have seen how these individual cellular marvels unite to form the breathtaking tapestry of multicellular life, and how understanding their delicate balance is key to comprehending health and disease. As we conclude this chapter, I invite you once more to pause, to breathe, and to reflect. Look at your own hand. Marvel at the living, breathing masterpiece it represents. Every cell, every tissue, every organ system is a testament to billions of years of evolution, a legacy of profound biological ingenuity. The deeper we peer into this unseen universe within us, the more we appreciate the extraordinary wonder of life itself and the immense potential that lies in unraveling its enduring mysteries. The cell, in its magnificent simplicity and bewildering complexity, remains the ultimate frontier, a living, breathing interface between the known and the eternally wondrous. And our exploration, dear friends, will continue. We have journeyed together, haven't we? Through the shimmering veil of the cell membrane, into the bustling metropolis of the cytoplasm, and across the intricate pathways of energy and information that define life itself. We have marveled at the nucleus, the silent architect, and the mitochondria, the tireless engines. We have witnessed the elegant dance of replication and the profound wisdom of differentiation, where a single cell's destiny unfolds into the intricate tapestry of a living being. And in our last shared moments, we confronted the delicate balance of homeostasis, the subtle symphony that, when disrupted, can lead to the discord of disease. But even as we explored the magnificent individual, the solitary cell, and then its grand orchestration into tissues and organs, there remains a layer of profound interaction, a silent, ceaseless conversation that stitches life together beyond mere signalling. We must now turn our gaze to the very fabric that binds cells, that provides them with context, and indeed influences their fate. It is a realm of exquisite detail, where cells do not merely exist next to each other, but with each other in an intimate, dynamic embrace. Imagine, if you will, that each cell is not just a building in a city, but an artisan, constantly shaping and being shaped by its immediate surroundings. This surrounding, this intricate micro-environment beyond the cell membrane, is not empty space. Oh no, it is a vibrant, complex tapestry known as the extracellular matrix, or ECM West. Picture a vast microscopic garden, meticulously cultivated by the cells themselves, yet in turn dictating how those cells grow, move, and communicate. The ECM is a marvel of biological engineering, a non-living network of secreted macromolecules that provide structural and biochemical support to the surrounding cells. It is the scaffolding, yes, but also the communication hub, the reservoir of growth factors, and the regulator of cellular behavior. It is a testament to the fact that no cell is truly an island. Consider its primary components, woven together with astonishing precision. First, there are the collagen fibers, strong and resilient, like the steel girders of our miniature city. These fibrous proteins are the most abundant proteins in mammals, providing tensile strength, preventing tissues from tearing. Imagine bundles of microscopic ropes, twisted and intertwined, giving skin its elasticity and bones their rigidity. Without collagen, our tissues would simply collapse, unable to withstand the constant forces of life. Then there is elastin, a protein that, as its name suggests, provides elasticity. Think of it as the spring in the mattress, allowing tissues like your skin, lungs, and blood vessels to stretch and recoil without losing their shape. It grants a supple resilience, a graceful give and take that allows our bodies to move and breathe with effortless grace. Interspersed within this fibrous network are the proteoglycans, complex molecules consisting of a protein core with long chains of sugars, glycosaminoglycans, attached. These are like sponges, attracting and retaining vast amounts of water, forming a hydrated gel-like substance that fills the spaces within the ECM. This gel acts as a shock absorber, resisting compressive forces, much like the jelly in a cushion protects its contents. It also creates a medium through which nutrients, waste products, and signaling molecules can diffuse, facilitating the constant dialogue between cells. And finally, we find of fibronectin and laminin, adhesive glycoproteins that act as molecular bridges. Fibronectin, for instance, links cells to collagen fibers and other ECM components, guiding cell migration and playing a crucial role in wound healing. Laminin, on the other hand, is a major component of the basal lamina, a specialized layer of ECM that underlies epithelial cells, providing a foundation upon which these cells can grow and organize. Together, these components form a dynamic, intelligent frontier. The ECM doesn't just hold cells in place, it actively instructs them. It can influence gene expression, cell proliferation, differentiation, and even cell survival. A cell senses the stiffness of its environment, the presence of specific binding sites, the chemical cues embedded within this intricate matrix, and it responds, adapting its behavior in a continuous feedback loop. It's a dialogue, a silent negotiation between the cell and its world. But how do cells physically connect to this remarkable matrix and to each other? This brings us to the fascinating world of cell adhesion molecules, or CAMs. These are specialized proteins on the cell surface that mediate cell-to-cell and cell-to-ECM interactions. They are the hands that reach out, the anchors that hold fast, and the antennae that sense the immediate environment. Consider the integrins, a family of transmembrane proteins that are crucial for connecting cells to the ECM. Imagine them as tiny grappling hooks extending from the cell membrane, binding to components like fibronectin and laminin in the ECM, but their role extends far beyond mere attachment. Integrins are two-way signaling transducers, They sense the external environment and transmit information into the cell, influencing its internal machinery, cytoskeleton, and gene expression. Conversely, internal signals can alter integrin activity, changing the cell's grip on its surroundings. This bidirectional communication is vital for processes like cell migration, wound healing, and tissue development. Then we have the molecules responsible for cell-to-cell adhesion, creating the cohesive structures we recognize as tissues. Cadherins, for example, are a major class of CAMs that mediate calcium-dependent cell-to-cell adhesion. They are like tiny zippers interlocking with cadherins on an adjacent cell, forming strong connections that hold epithelial cells together, allowing them to form continuous sheets. There are different types of cadherins, each expressed in specific tissues, contributing to the selective sorting and organization of cells during development. Beyond individual adhesion molecules, cells also form specialized structures called cell junctions, which are like reinforced seams in the fabric of a tissue. We can broadly categorize them into three main types, each serving a distinct purpose. First, the tight junctions, or zonular occludens. These are like an impenetrable seal, forming a continuous band around the cell, literally fusing the outer layers of adjacent cell membranes. Imagine the waterproof seal of a Ziploc bag. They prevent the passage of molecules and ions through the space between cells, creating a barrier that separates fluid compartments. Think of the lining of your gut or the blood-brain barrier. Tight junctions are indispensable for maintaining these critical physiological barriers, ensuring that substances pass through cells where they can be regulated rather than bypassing them. Next, the yesosomes, or macular adherens. These are like spot welds, providing strong mechanical attachments between cells. Picture rivets holding two metal plates together. They link the intermediate filaments of adjacent cells, distributing mechanical stress across a network of cells. Tissues subjected to significant mechanical stress, such as your skin or heart muscle, are rich in desmosomes, ensuring their integrity and preventing them from tearing apart under tension. And finally, the gap junctions, or nexus. These are perhaps the most intimate of connections, functioning as direct channels of communication between adjacent cells. Imagine tiny tunnels or pores, allowing small molecules, ions, and electrical signals to pass directly from the cytoplasm of one cell to another. They facilitate rapid intercellular communication, coordinating the activities of groups of cells. In your heart muscle, for instance, gap junctions ensure that electrical signals spread quickly, allowing heart cells to contract in a synchronized rhythm. In developing embryos, they allow cells to share signaling molecules, coordinating growth and differentiation. These intricate networks of ECM, CAMS, and cell junctions are not static structures. They are constantly being remodeled, broken down, and rebuilt in response to cellular needs and environmental cues. They are the silent orchestrators of tissue architecture, allowing for the incredible complexity and adaptability of multicellular life. And this profound understanding of cellular interaction and the intrinsic wisdom of the cell itself leads us to perhaps one of the most exciting and hopeful frontiers in cell biology: regenerative medicine and the power of stem cells. Remember our discussion of cellular differentiation, how a single fertilized egg gives rise to all the specialized cell types in your body? At the heart of this process, And indeed, at the heart of repair and renewal throughout life are stem cells. These remarkable cells possess two defining characteristics. They can self-renew, divide to produce more stem cells, and they can differentiate, specialize into various cell types. Imagine them as the master keys to the body's repair shop, holding the potential to become almost any cell type. We speak of pluripotent stem cells, like embryonic stem cells, which have the potential to differentiate into any cell type of the body. And we have multipotent stem cells, like adult stem cells found in bone marrow or fat tissue, which can differentiate into a more limited range of cell types within a specific lineage. The promise of stem cell research is immense. What if we could harness this inherent ability to repair damaged tissues and organs? What if we could grow new heart muscle after a heart attack, or replace neurons lost in neurodegenerative diseases like Parkinson's or Alzheimer's? This is the vision of regenerative medicine, using cells either from the patient's own body or from carefully cultivated sources to restore normal function to diseased or damaged tissues. This field extends into tissue engineering, where scientists are learning to create functional tissues and even organs in vitro outside the body. Picture scaffolding, often biocompatible materials mimicking the natural ECM, seeded with stem cells or specific differentiated cells. These cells are then encouraged to grow, differentiate, and organize into complex structures, guided by growth factors and mechanical stimuli, much like they would in the body. Imagine growing a new piece of skin for burn victims, or even a functional bladder or trachea. The possibilities are truly transformative, and this journey doesn't end there. Our ever-deepening understanding of the genetic blueprint, the very language of life we explored in Chapter 5, has opened doors to genetic engineering and personalized medicine. With tools like CRISPR-Cas9, we are learning to edit genes with unprecedented precision, correcting mutations that cause diseases. Imagine a future where genetic disorders are not merely managed but cured by directly repairing the faulty instructions within a patient's own cells. This is the essence of personalized medicine, tailoring treatments to an individual's unique genetic makeup, predicting disease susceptibility, and developing therapies that are most effective for you. It is the ultimate expression of our journey through cell biology, taking the foundational knowledge of how cells work and applying it to heal, to restore, and to extend the quality of life. So, as we bring this foundational lecture to a close, let us pause once more to reflect. We began by peering into an unseen universe, a complexity that rivals galaxies hidden within us. We have traversed its vastness, from its protective outer membrane to its deepest genetic secrets, from its tireless energy production to its intricate communication networks. We have seen how individual cells, through profound cooperation and constant interaction, construct the magnificent architecture of life. The journey of discovery in cell biology is far from over. Indeed, each answer only illuminates more questions. Each solved mystery reveals new layers of wonder. The cell remains a dynamic, intelligent frontier, ceaselessly active, endlessly adapting, and holding within its shimmering boundaries the very essence of existence. May your curiosity remain boundless, your wonder unwavering, and your appreciation for the intricate beauty of life at its most fundamental level forever deepened. The universe within you and within every living thing continues its silent, majestic dance. Keep listening, keep observing, keep marveling, for the adventure truly has only just begun. The journey through the intricate dance of cells and their environment, exploring the profound potential of regenerative medicine and genetic engineering, brings us to a new precipice. We have peered into the very essence of life, understanding how its fundamental units collaborate, communicate, and self-repair. Yet, the story of the cell is not merely one of observation and repair. It is also one of inspiration, of imagination, and of the audacious endeavour to understand and perhaps even to design life itself. As we drift further into the contemplative spaces of our dreamy academy, let us now cast our gaze not just upon the cell as it is, but upon the cell as it might be, and how our ever-deepening understanding empowers us to consider possibilities once confined to the realm of science fiction. We have, together, unfolded the understanding of the cell like origami in reverse, revealing its elegant internal mechanisms, its metabolic ballet, its genetic poetry, and its harmonious existence within the grand symphony of multicellular life. We have seen how cells, through their intricate adhesion molecules and junctions, weave the very fabric of our being, creating a tapestry of interconnectedness that extends from the smallest protein to the vastness of an organ system. Now, we stand at a threshold where the knowledge gleaned from centuries of patient observation and groundbreaking discovery begins to converge with the tools of deliberate creation. Imagine, if you will, that having meticulously studied the blueprint and the functioning of a magnificent self-assembling, self-replicating machine, we are now learning to read its living code with such fluency that we might begin to write new verses, new chapters, or even entirely new sagas within the epic of life. This, my friends, is the heart of synthetic biology, an emerging frontier that seeks not merely to understand existing biological systems, but to design and construct new ones, or to re-engineer existing ones for novel purposes. Think back to the cell as a miniature city, bustling with specialized districts and interconnected transport networks. We have meticulously mapped its roads, identified its factories, and understood its energy plants. Synthetic biology proposes that, with this deep comprehension, we can begin to draw new city plans. Perhaps we can reroute a metabolic pathway to produce a new medicine more efficiently, or design a cell that can detect and neutralize pollutants in the environment, or even engineer microbes that can generate sustainable biofuels. It is an ambition that resonates with the very spirit of creation, a profound leap from observation to guided intervention. At its core, synthetic biology relies on the modularity of biological systems. We have seen how genes are like discrete instructions, how proteins are specific molecular machines, and how pathways are interconnected sequences of biochemical reactions. The brilliance of synthetic biology lies in treating these biological components as standardized parts, much like electronic components that can be assembled into complex circuits. Researchers are building libraries of these biobricks, genetic switches, protein sensors, metabolic modules. With these standardized parts, they can then design and construct novel genetic circuits, orchestrating cellular behavior in unprecedented ways. Consider the elegance of this approach. We are not just tinkering, we are learning the grammar of life itself to compose new sentences, new paragraphs, Imagine engineering a yeast cell, for instance, not just to ferment sugars into alcohol, but to produce complex pharmaceuticals like artemisinin, an anti-malarial drug, far more sustainably and affordably than traditional chemical synthesis. Or envision bacteria programmed to enter tumor cells and deliver therapeutic agents directly, minimizing side effects on healthy tissues. These are not distant dreams. Many such projects are already underway, transforming our approach to medicine, energy, and environmental stewardship. This endeavor requires a multidisciplinary dance, a harmonious collaboration between molecular biologists, geneticists, biochemists, and engineers, all speaking the common language of the cell. It's a testament to the integrated nature of scientific inquiry, where the insights gained from understanding a single protein's fold can cascade into the design of an entirely new biological function. We are learning to program cells not with binary code, but with the elegant four-letter alphabet of DNA, rewriting genetic instructions to achieve specific beneficial outcomes. Yet, as with any power, this capacity for deliberate biological design brings with it a profound responsibility and a host of ethical considerations. When we begin to engineer life at its fundamental level, we touch upon questions that extend far beyond the laboratory bench. What are the boundaries of intervention? How do we ensure safety, preventing unintended consequences when introducing engineered organisms into complex ecosystems? What are the societal implications of designing organisms for specific purposes, and how do we ensure equitable access to these powerful new technologies? These are not questions that can be answered solely by scientists. They require a broader societal dialogue, a shared contemplation that engages ethicists, philosophers, policymakers, and the public. We are, in essence, becoming co-authors in the ongoing story of life on Earth, and with that privilege comes the solemn duty to proceed with wisdom, foresight, and a deep respect for the intricate balance of the natural world. The philosophical implications are vast. What defines natural when we can design biological systems? How does our ability to engineer life reshape our understanding of evolution, of purpose, and of our place within the biological tapestry? These are not simple inquiries, but rather invitations to deep reflection, urging us to consider the profound interconnectedness of all living things and the delicate responsibilities that come with such knowledge. Even as we push these frontiers, it is crucial to remember that the cell, in all its perceived simplicity and astonishing complexity, remains an endless source of wonder and an ongoing mystery. For every answer we uncover, a dozen new questions bloom, inviting us deeper into its hidden dimensions. We may learn to manipulate its genes, to re-engineer its pathways, but the sheer elegance of its self-organization, its inherent resilience, and its capacity for adaptation continue to humble us. The cell is a dynamic, intelligent frontier, ceaselessly evolving, ceaselessly surprising. The journey we have undertaken together, from the shimmering boundary of the cell membrane to the bustling cytoplasm, from the genetic blueprint of the nucleus to the energetic pulse of the mitochondria, from the intricate dance of communication to the grand design of multicellularity, and now to the ambitious horizons of synthetic biology, is far from over. It is a testament to the boundless curiosity of the human spirit, a relentless quest to understand the very fabric of existence. And as we conclude this particular chapter of our shared exploration, I invite you to carry this sense of wonder this profound appreciation for the microscopic universe with you. Look at your own hand. Consider the atoms in your bones, the cells in your skin, the neurons firing in your brain. Each an intricate tapestry, a miniature city, a living, breathing interface with the world. You are, in every fibre of your being, a testament to the astonishing power and beauty of cell biology. The elegance of life, in its myriad forms, begins with the cell. It is the silent, tireless architect of our existence, a testament to complexity that rivals galaxies, a universe hidden within us, waiting always for curious minds to explore its depths. And so, the lecture concludes, but the journey of discovery for each of us continues. May your own cells carry you forward, ever curious, ever wondrous. Our journey through the intricate tapestry of cellular existence, you will recall, culminated in a contemplation of synthetic biology. The audacious ambition to design, to construct, to re-engineer life itself. It is a frontier that beckons with immense promise, yet simultaneously casts long shadows of ethical inquiry, challenging us to wield this nascent power with profound wisdom and an unwavering sense of responsibility. We stand at the precipice of not just understanding life, but perhaps in some rudimentary ways, writing new chapters of its story. But even as we gaze upon this horizon of engineered possibility, we must acknowledge the boundless expanse of what remains unseen, unheard, and utterly unknown. For all our sophisticated instruments, our burgeoning knowledge, and our ingenious designs, the cell still holds secrets that humble us, mysteries that invite us to look deeper, to listen more intently to the whispered wisdom of existence itself. Imagine, if you will, that the cell, this miniature city we have explored, is not merely a collection of organelles and pathways, but a living poem, an ongoing symphony of processes so exquisitely coordinated that they defy our most complex algorithms. We have dissected its verses, identified its instruments, and even begun to compose our own brief melodies within its grand score. Yet, the true genius of the composition, the ineffable spark that transforms mere molecular machinery into vibrant, self-aware life, often eludes our grasp. Consider the phenomenon of emergence. We have spoken of individual molecules, of proteins folding into precise shapes, of membranes forming selective barriers. But when these myriad components interact in specific ways, something utterly new and unpredictable arises. A single neuron is a fascinating entity, but billions of neurons, interconnected in a vast network, give rise to consciousness, to thought, to dreams, to the very capacity for self-reflection that allows us to ponder these mysteries. This is emergence, the whole becoming greater and fundamentally different than the sum of its parts. It is the magic woven into the fabric of the cosmos, from the formation of galaxies from dust to the flowering of a single cell into a sentient being. How does a collection of non-living molecules coalesce to form a living entity? What is that qualitative leap, that threshold crossed, where the chemical reactions become life? This is a question that transcends the purely biological, venturing into the realms of philosophy and even metaphysics. We can describe the conditions, the energy gradients, the self-replicating polymers, but the essence of the transition, the initial spark, remains one of the universe's most profound and beautiful enigmas. We are, in a sense, trying to understand the genesis of a whisper that became the roar of existence. And what of the cell's incredible adaptability, its resilience, its capacity for self-repair and self-regulation even under duress. We understand the mechanisms, the DNA repair enzymes, the stress response pathways, the intricate feedback loops that maintain homeostasis. But the sheer will of a cell to persist, to thrive, to navigate an ever-changing environment speaks to an inherent intelligence that extends beyond mere programmatic responses. It is a quiet, persistent dance with entropy, a ceaseless effort to maintain order against the universe's natural inclination towards chaos. We observe cells communicating, sensing their environment, making decisions, even dying in a programmed, altruistic manner for the good of the larger organism. These are not passive, inert sacks of chemicals. They are active, dynamic agents, engaged in a continuous dialogue with their surroundings. How much of this cellular intelligence is hardwired, and how much is a flexible, adaptive learning process occurring at scales we are only beginning to fathom? The more we learn, the more we realize the depth of the cell's sophisticated internal world, a universe of computation and interaction happening at speeds and scales that dwarf our most advanced supercomputers. Our journey has also illuminated the profound interconnectedness of all life, from the single-celled ancestor that gave rise to the breathtaking diversity we see today, to the intricate symbiosis within our own bodies. Where bacterial cells outnumber our human cells, life is a grand, collaborative project. The air we breathe, the food we eat, the very ground beneath our feet, all are products of and participants in this vast, interwoven web of cellular activity. When we look at a cell, we are not merely looking at a discrete unit. We are glimpsing a microcosm of the entire biosphere, a reflection of the universal principles that govern existence. This perspective invites a deep sense of humility. For all our advancements, we remain but curious observers, privileged to witness the unfolding miracle of life. The knowledge we gain, the technologies we develop, must always be tempered with reverence for the inherent wisdom of nature, a wisdom honed over billions of years of trial and error, of adaptation and evolution. When we venture into synthetic biology, we are not just playing with molecules, we are engaging with fundamental forces that have shaped life since its inception. The ethical considerations we touched upon in our last session thus become not just constraints, but guiding stars. They remind us that power without wisdom is peril, that scientific endeavor divorced from a deep respect for life's intrinsic value can lead us astray. Our capacity to alter the fundamental building blocks of life carries with it an immense responsibility, to ensure that our innovations serve to enhance life, to heal, to understand, rather than to diminish, to harm, or to exploit. What, then, is the ultimate purpose of this slow, deliberate journey into the heart of the cell? Is it merely to accumulate facts, to master techniques, to unlock new medical breakthroughs? While these are indeed noble pursuits, I propose that the deeper purpose lies in something far more profound. It is to cultivate a deeper sense of wonder, to foster a more intimate connection with the very essence of our own being, and to awaken within us a profound appreciation for the miraculous ordinary that surrounds us. When you look at your own hand, you're not merely seeing skin and bone. You're witnessing billions of cells, each a bustling metropolis, each meticulously performing its role in a symphony of life that has been playing for eons. You're a testament to cellular resilience, to genetic inheritance, to metabolic genius. You are, in every fibre of your being, a living, breathing monument to the extraordinary power and beauty of the cell. Our exploration of cell biology fundamentals is not an endpoint, but an invitation. It is an invitation to remain curious, to question, to marvel, and to continue seeking understanding not just in laboratories, but in the quiet contemplation of a leaf unfurling, a child growing, or the simple act of breathing. For the universe, in its infinite wisdom, has chosen to express its most fundamental truths not in grand celestial displays alone, but also in the delicate dance of proteins, the silent replication of DNA, and the shimmering boundary of a cell membrane. The journey of discovery is an endless one, a winding path that continually reveals new vistas, new questions, and new depths of understanding. We have only just begun to scratch the surface of the cell's infinite complexity, to decipher a few lines from its ancient ongoing narrative. But what we have learned, what we have seen, is enough to fill us with an abiding sense of awe, a reverence for the miniature universes that reside within us, that comprise us, and that connect us all. Let us carry this wonder forward, allowing it to inform our choices, to inspire our actions, and to guide our continued exploration of the most fundamental yet most profound aspects of existence. For in the heart of every cell lies a secret waiting to be rediscovered, a truth waiting to be embraced, a connection waiting to be made. The lecture, in its formal sense, may draw to a close, but the learning, the marvelling, the profound connection to life itself has only just begun. The unseen universe continues to unfold, and we, with open minds and hearts, remain its eager, lifelong students. We have journeyed far, haven't we? From the shimmering boundary of the cell membrane to the intricate dance of genetic expression, from the relentless hum of metabolic factories to the profound ballet of cellular communication. Yet, as with any grand exploration, the deeper we delve, the more vast and wondrous the unknown becomes. Our final steps together will lead us not to definitive answers, but to the very edge of our current understanding, to the profound and humbling mysteries that still enshroud the cellular realm. Let us speak then of Ga, emergence. Imagine, if you will, a single note played on a piano. It is a sound, yes, distinct and pure, but combine it with others in sequence and harmony, and suddenly a symphony arises. The symphony is more than the sum of its notes. It possesses qualities, emotion, narrative, grandeur, that are entirely new, emergent properties born from the intricate arrangement and interaction of simpler components. This, my friends, is the essence of emergence, and it is a phenomenon woven into the very fabric of life, from the smallest cell to the most complex organism, and indeed to the cosmos itself. Within the cell we witness emergence at every scale. Individual proteins, each a marvel of molecular engineering, possess specific functions, but when these proteins gather, fold and interact within the cytoplasm, they form complex molecular machines, ribosomes, motor proteins, enzymes, whose collective action gives rise to the cell's dynamic capabilities. Consider the mitochondria, that powerhouse we explored. Its inner membrane, a labyrinthine landscape, is studded with countless protein complexes. Each complex performs a specific step in oxidative phosphorylation, but it is their collective, orchestrated activity that generates the vast quantities of ATP, fueling the entire cellular enterprise. The energy of life itself emerges from this molecular symphony, and then ascend another layer. Individual organelles, each with its designated role, operate in concert. The nucleus commands, the ER synthesizes, the Golgi packages, the mitochondria energize. Yet the living cell, that self-sustaining, self-regulating, adapting entity, is not merely a collection of these parts. It is a new entity entirely, with properties like growth, reproduction, and responsiveness that do not reside in any single organelle, but emerge from their integrated, ceaseless interaction. A cell, in its entirety, is an emergent phenomenon, a miniature universe born from the intricate interplay of its components. Now extend this vision further still. Consider the journey from single-celled life to the magnificent tapestry of multicellular organisms. Billions upon billions of cells, each a self-contained marvel, come together. They don't just aggregate, they differentiate, specialize, communicate, and cooperate in ways that transcend the capacities of any individual cell. From this coordinated dance, tissues emerge, then organs, then organ systems, and finally an organism, a human being, a soaring eagle, a majestic redwood. And within that organism, particularly within the human brain, we encounter perhaps the most profound emergent property of all, consciousness, the subjective experience of you, your thoughts, your emotions, your sense of self. These do not reside in a single neuron, nor in a simple cluster of neurons. They emerge miraculously from the intricate, dynamic, electrochemical symphony played by trillions of neural connections. The very act of wonder we share now, the contemplation of these profound truths, is itself an emergent property of the biological complexity within us. This leads us to another profound mystery: cellular intelligence. We have spoken of cells responding to signals, making choices, adapting to their environment. But is this merely a sophisticated form of biochemical reflex, or is there something more akin to rudimentary cognition at play? We observe cells navigating complex environments, sensing gradients of chemicals, remembering past encounters, even communicating distress or cooperation across vast distances within a tissue. A fibroblast, for instance, will migrate towards a wound site, guided by a complex interplay of signals, adhering to the extracellular matrix and initiating repair. Is this merely a programmed response, or does it involve a form of cellular decision-making, an interpretation of its world? Consider the immune cell, a sentinel patrolling the bloodstream. It encounters a foreign pathogen. It doesn't just react, it identifies, assesses the threat, mounts a targeted response, and often quotes 19 that pathogen for future encounters. This capacity for memory, for adaptive learning, for processing vast amounts of information and initiating appropriate, often novel responses, challenges our simplistic notions of what, quote, 20 truly means. Perhaps intelligence is not solely the domain of complex brains, but exists in myriad forms, scaled and expressed differently across the biological spectrum. Perhaps the cell, in its intricate dance of molecular recognition and signal transduction, possesses a form of distributed cognition, a wisdom encoded in its very structure and dynamics. It is a question that invites humility, urging us to reconsider our anthropocentric definitions and to marvel at the profound adaptive capacities of life at its most fundamental level. And finally, my friends, let us reflect upon the interconnectedness of all life, a truth made profoundly clear by the study of the cell. Look at your own hand, the intricate tapestry of skin, muscle, bone, and nerve. Every cell within it, from the deepest marrow to the outermost epidermal layer, shares a common molecular language. The DNA in your nucleus, the ribosomes building your proteins, the mitochondria generating your energy, these are not unique to you. They are variations on a theme, echoes of a grand evolutionary symphony played out over billions of years. We share this fundamental molecular toolkit with every living thing on Earth. The genetic code, the elegant double helix, is almost universally conserved. The mechanisms of protein synthesis, the very ATP that powers your thoughts, are found in the simplest bacterium and the tallest tree. This profound universality speaks to a common ancestry, a single, ancient origin from which all life has diverged. We are, in a very real sense, cousins to the fungi, siblings to the plants, and distant relatives to the archaea flourishing in the deepest vents of the ocean. The boundary between quote 21 quote and quote 22 quote, while crucial for individual identity, dissolves into a continuum when viewed through the lens of deep evolutionary time. The very air you breathe, the water you drink, the food that sustains you, all are products of cellular life. Photosynthesis, the miraculous process by which plants and algae convert sunlight into energy, underpins almost all life on Earth, producing the oxygen we inhale and the organic molecules we consume. The delicate balance of ecosystems, the cycling of nutrients, the health of our planet, all are expressions of the collective activities of trillions upon trillions of cells, from the microscopic plankton in the oceans to the towering giants of the rainforest. We are not isolated entities, but integral threads in an immense, living tapestry, interconnected at every scale, from the subatomic to the planetary. As we reach the culmination of our journey through the quote 23, it is with a sense of both profound understanding and even deeper wonder. We have peeled back layers of complexity, marvelled at the elegance of biological design, and glimpsed the ceaseless activity that defines life. Yet the cell continues to hold vast, unresolved mysteries. How exactly does life first arise from non-living matter? What are the full extent of the regulatory networks that govern cellular behaviour, and how do they achieve such robust adaptability? What is the precise nature of the quote 24 that allows a single fertilized egg to develop into a complex conscious being? These are not mere gaps in our knowledge. They are invitations to future discovery, beckoning us to continue the grand quest for understanding. The field of cell biology, far from being a static repository of facts, remains a vibrant, dynamic frontier. New technologies, from advanced microscopy that allows us to visualize molecular dances in real time, to sophisticated genetic editing tools that offer unprecedented control, are continually pushing the boundaries of what we can see, understand, and even manipulate. The ethical considerations surrounding these advancements will continue to shape our discourse, challenging us to wield this knowledge with wisdom, humility, and a profound respect for the intricate balance of life. Our exploration has been a gentle reminder that the grandest wonders are often found not in the distant cosmos, but in the intimate universe hidden within us, within every leaf, every drop of water, every breath. It is a realm of profound, intricate beauty, a testament to the inherent wisdom of existence at its most fundamental level. I'd love for you to carry this sense of wonder forward. Let the knowledge we've shared not be an endpoint, but a beginning, Let it ignite a deeper curiosity, a greater reverence for life in all its forms, and an appreciation for the astonishing complexity that defines our very being. The cell, that miniature city, that living, breathing interface, is a constant source of inspiration, a silent teacher whispering truths about connection, resilience, and the endless possibility of life. May your journey of discovery continue, ever illuminated by the light of scientific inquiry and the warmth of an open, wondering heart. Thank you for embarking on this slow and thoughtful adventure with me. The cells within you, within all of us, continue their tireless, beautiful work, a constant reminder of the profound, intimate, universal truth that we are, each of us, a living, breathing miracle,